So this is the work that I have started in 2017 with the funding offered by the Leukemia UK charity. So at that time I was awarded the John Goldman Fellowship to basically do something to carry out a new project to try to understand the molecular mechanism involved in this leukemia, KMT2A-rearranged. So KMT2A is an oncogene that is basically involved in chromosomal translocation that affect roughly 10% of patients with acute myeloid leukemia. And is actually very common in children with acute lymphoblastic leukemia. So over 90% of children that are diagnosed before reaching 1 year of age are affected by this leukemia. And this is particularly a particularly aggressive form of leukemia that is also chemoresistant. Now, we basically know that despite there has been lots of research on new therapy for acute myeloid leukemia, patients are mostly still treated with chemotherapy and bone marrow transplantation. And we know that some patients with specific genetic mutation, chromosomal rearrangement, will do okay with this treatment, and some of them that actually are quite a significant percentage will relapse, which means the disease will come back. And KMT2A rearrangement fit into this group, so the patients actually do not really do well with this chemotherapy treatment and relapse. Now, when I basically started this project, I was actually quite intrigued by the fact that apart from having these chromosomal translocations, patient with KMT2A rearrangement seem also to have mutations in genes belonging to signaling pathways, so RAS, MAP kinases, and also even if these genes were not found mutated in these patients, they were consistently reported as active kinases, active proteins, and those are important for the proliferation of the leukemic cells as well as to resistance to treatment. So we basically thought about trying to understand what was the functional meaning of this. So how these signaling pathways could contribute to the leukemia, and what was controlling these signaling pathways. And this is how we started our research on phosphatase PP2A. So this phosphatase is basically an enzyme very important for controlling phosphosignaling. And before our before we started our research, it was already reported as functionally inactivated in leukemia patients, including AML. So what we present in this poster is a characterization of the expression of SET, which is one of the PP2A endogenous inhibitor. So what we found is basically that this gene encoding for the protein SET is overexpressed across AML patients, including those with KMT2A rearrangement. And the expression of SET is actually highly correlates with the worst prognosis in acute myeloid leukemia patients, meaning that those patients that have high expression of SET will actually tend to survive less. And what we found extremely interesting was the fact that the expression of SET also correlates with the expression of genes such as OXA5, OXA9, OXA10, MACE, that are, the OXA genes are part of the leukemic stem cell genetic signature in acute myeloid leukemia. And also these genes are upregulated by KMT2A chromosomal rearrangement. So we thought, okay, so this might actually have a functional meaning. So what happens if we knock down, if we silence the expression of SET in leukemic cells? And we found that indeed those leukemic cells expressing the KMT2A rearrangement are extremely dependent on SET expression. And when we knock down the expression of SET by using RNA interference, those cells can actually survive for not very long in our lab. And this is something that seems to be very specific to KMT2A rearrangement. So these are some of the data that I will present in my poster in BSH. And currently we are looking for funding to actually continue this project, so to understand a little bit better why these leukemic cells are so dependent on SET, what is the molecular pathways that are affected, and even if there are maybe other subgroups of acute myeloid leukemia that might show the same dependency. Another important aspect that we cover in this poster is potential pharmacological strategies. What I mean is obviously for a biologist, it's very nice to find a gene that you can knock down in the lab and that kills the leukemic cells. But we know that we can't do this in patients. We can't knock down genes in human beings. So we thought, okay, this is very nice, but how are we gonna ever translate this into a treatment for patients? We were lucky that we found a pharmacological a drug that is called FTY-720 that is already FDA approved for multiple sclerosis, so a completely different indication, and in the scientific literature was reported as an inhibitor of SET PP2A interaction. So we thought, okay, let's maybe try to see what's the effect of this inhibitor in the leukemic cells. And again, we basically found that this inhibitor can arrest the proliferation of the leukemic cells and can also increase the sensitivity of the PDX, which are patient-derived xenotransplants, so a very good model of leukemia, to standard chemotherapy. So this is all good news, but unfortunately, because FTY-720 has a very strong immunosuppressive profile, it cannot be repurposed to AML patients, meaning that we definitely need to understand how we can pharmacologically target SET without inducing side effects such as the immunosuppression that would be not tolerable for acute myeloid leukemia patients. So this is where we are and basically where we want to go.